All right, so now you have an understanding about what organic chemistry is, inorganic chemistry. Now you understand about how carbons in organic chemistry can be formed as chains or they can be rings. Now you can understand that carbon can be bound by 4 different bonds and you can make big structures. Now you can understand that when we break things, you could have energy that gets released. There's so much now we just went into. Now the only thing that we really have to cover left about chemistry is, well, what are these structures that we've been talking about? We learned about which different types of structures, but what structures are you talking about? Well, it's the stuff that we've been talking about, or that you know about, just foods that you're eating. So let's go into that, all right? So we have these big structures. Now, they're called biological macromolecules. Macro meaning big as opposed to micro being small. But it's kind of a weird thing because even though it's macromolecules, you still can't see with your naked eye, you know what I'm saying? But these are just bigger than just 2 carbons. These are much bigger. And they all contain some sort of carbon skeleton. So we have 4 biological macromolecules. 4 of them. They all contain carbon. You've heard of them. Proteins. And they give you 4 calories per gram. I'm not going to ask you about that, but if you're interested, you're in nutrition and things, just give me a heads up about that. But I won't ask you about what they give off. We also have carbohydrates, which are better known as sugars. We have fats. And they give off a whopping 9 calories per gram. These are the wasted calories. When you look at how much fat is in Snickers, you'll see it's a lot there. And it just, you'll have the total calories altogether as, let's say, maybe 400 calories, but 300 of those calories come from fat. And the real reason why people are fat or obese is not because of a thyroid problem that people go, oh, I got a thyroid problem, that's why. Meanwhile, they're still eating, right? It's not that. It's just simply the amount of input doesn't equal the amount of output. The amount of calories that you're taking in by your food, let's say it's 3,000 calories you're taking in of all the food you're eating that day, but the thing you're doing with exercise and walking, you're only burning off 1,000 of those calories. That leaves 2,000 calories not accounted for, so that turns into storage or fat. Does that make sense? Ideally, what you want is, 1,800 calories in, 1,800 calories out. That's it. But there is things with thyroid and estrogen and testosterone and growth hormone. There's a lot of other things that will break up things, and that does have a different rate in different people. And you just need to know what that is. You could have a slow thyroid, and not disease, I'm just saying a slower thyroid that actually deals with metabolism, but you know your own body, so you know don't eat that much food or exercise more, and that's where that happens. But that's the real reason, is the input of calories doesn't equal the output of calories, all right? And it seems like, not you guys, but it seems like when kids are in school and in college and they're really studious and they're studying, they're on their bum more than they are walking around the block or whatever. So we tend to do that, right? Because we're studying. Our noses are in the books. And really the main reason why people eat, besides energy, if you think about it, you're bored. Right? Think of the times that you go and grab that bag of potato chips. You're studying. I don't take it personal. I know studying's boring. Okay? You're watching TV and it comes to a commercial. Right? You're bored. So you go get something, right? That's what I'm saying. The most common reason why people eat inappropriately is because you're bored. Right? So if you know about that, then find other things to do while you're bored. Do some sit-ups during a commercial. Oh, I'm not going to do that. It's extra effort, right? I'm not going to do that, right? Yeah. But that's what it comes down to. So if you're aware of these, then that's fine. Then you just have to adjust it. You know it's bad for you to be getting a bag of potato chips, and you just control it. That's all. It's easier said than done. You know, and let's face it, you can't just have, you know, one Dorito. You got to eat the whole bag of Doritos, right? That kind of stuff. All right, and so on this, so you have proteins, carbohydrates, fats, and something called nucleotides, ATP, and nucleic acid. And the energy that comes out it varies, and we'll talk about that very briefly too. So let's talk about proteins first, okay? And the building blocks, the things that make up proteins, is amino acids. That's an amino acid. An amino acid has this alpha carbon in the center with a hydrogen, and we've got functional groups. We have an amino group on one side over here, and a carboxylic acid group on this side over here. There is 20 amino acids in our body, and all 20 amino acids have the same exact structure. Only thing different is the radical or the R group. This part here. This might just be a hydrogen. It might be a thiol, meaning like sulfur on it. It might be an alcohol on there. It might be a ring. That's what makes this all different. And of these 20 amino acids, they make up proteins. And there is a whole slew, thousands and thousands and thousands of proteins all in our body, just made by those 20 amino acids. Just think of it like all the words in the dictionary are made from just 26 letters of the alphabet. It's the same thing. So that's an amino acid. There's 20 different amino acids in our body, all identical except for that R group. And all are used to make up these, what we call proteins in our body. Long chains, they're also referred to as polymers. Anything that's a long chain is polymers. And they're just amino acids, all chained together, like pearls on a necklace. We have, and I abbreviate amino acids as AA, but we have essential amino acids. These are there's certain ones, 9 amino acids of the 20, that our bodies cannot make. We have to get it in our diet. So they're essential. Then we have non-essential, and there are 11 amino acids that our body knows how to make. They take parts of things and puts them together and stuff. These are your 20 amino acids. You've got to draw them all out on your test. No, you don't. And you don't have to recognize, I'll be honest with you, you don't have to recognize them. Okay? It's just too much. There's courses like biochemistry and stuff. I won't ask you, they have different names, aspartic acid, glutamic acid, stearine and stuff. I won't ask you about them. Just understand what an amino acid is. You should know what the basic things, what the identical parts are. You should identify that, but the R groups, no. You do if you need to go to biochemistry or medicine or whatever, nursing, yes. Pharmaceuticals, yes. You need to know all those and know which ones are polar, which ones are non-polar, which ones are charged. I'm not going to ask you that. It's just too much to do. I have other stuff I can really get you with. Okay? This is just in one ear, out the other. It's just too much. But you do need to know that there's 20. You do know that there's what essential and non-essential are, how many there are. You should know what's in yellow. The yellow parts over here is what they're all going to be the same, the amine and the carboxylic acid group on the alpha carbon. But the blue sections is what I'm trying to show you is all the R groups. And that's all different. That's what makes all the amino acids different. okay? And they all have different names. I'll mention the certain names of amino acids as we go along when we show you how to make a protein and stuff, but I'm not going to hold you to say which one is which. It's way over. Yeah. The yellow is, let me go back to this picture here. Okay. This is kind of reversed. In other words, this is the yellow that's showing on there. The blue is this. So it's upside down. Does that make sense? So on here, on this one, yellow here, blue here. But on this picture here, all of these are showing the yellow on the bottom and the blue up on top. Okay? And you'll see when I know in the back, you really can't see it in the back, but when you see, I'll blow it up when I do the video so you can see them. But yeah, all the yellow up there is the same exact structure on all 20 here. The blue, which is the R group, is different. And I'm just trying to be complete and just showing you that, but I wouldn't expect you to know those blue groups way too much. Okay? I won't have you memorize this, but I'm just trying to show you again the essential amino acids and the non-essential amino acids. And you'll see words like alanine and proline and stuff. Why they're equal over there? Well, it's trying to show you also in, you have arginine and histidine, which are essential for children but not for adults. Our bodies become more complex and can make those on there, okay? But you won't, again, you just need to understand what an essential and a non-essential amino acid is. Don't worry about what ones they are. All right, now I'm not going to do this part now. We're going to deal with this actually towards the end on making a protein. But just trying to show you how amino acids are building blocks of proteins, we'll learn that this green thing here is inside the cell. It's called a ribosome. And these things here, they're called transfer RNA. They carry amino acids. So don't worry too much about this. Just trying to orient you. But this thing is carrying an amino acid. It's going to attach to something that has a long chain of amino acids, a protein. So this amino acid is going to attach to this one. And then this transfer RNA is going to fly off of there without the amino acid. And then another transfer RNA with amino acid comes down and attaches to that and goes off. It's like an assembly line that goes on. Okay, this is called translation. We'll get into that later. But I'm just emphasizing that amino acids are the building blocks, the pearls on the necklace, the building blocks of a protein, a long chain of amino acids. So how do these amino acids attach to each other? Well, it's a covalent bond. And it is what we call a specific type of covalent bond called a peptide bond. Okay? So a peptide bond is a covalent bond attaching amino acids together. Okay? And that's what that is. And it joins the amino group of one amino acid to the carboxylic acid group of another amino acid. And it's formed through these synthesis, which is a dehydration reaction. Okay? Nomenclature. Fancy word about how to name things. And it's pretty easy, right? A dipeptide is 2 amino acids. A tripeptide is 3 amino acids. Oligo means a small amount, 4 to 15. A polypeptide is from 16 to 15 amino acids. Anything greater than 15 amino acids, we simply call a protein. So that's how we name things. Okay? So what can proteins do? Why are we making a big thing about proteins? Well, there's a lot of things to do. Structure. The keratin, the hard stuff that's in your nails, in your skin, in your hair. Keratin, that's a protein. Collagen, all the tissue in your liver, the stroma, the substance of your liver, and the substance in the walls of the stomach, and the walls of, let's say, the heart, and your muscle. That's all what we call collagen. That's protein also. It can communicate. We've been mentioning a couple things about hormones, like estrogen and progesterone and testosterone. Well, actually, those are the wrong ones, because those are steroids. But there's certain hormones that are proteins. So they're going to attach to receptors. A key is a hormone. A keyhole is a receptor. The key is made of a protein. The keyhole is made of a protein, the receptor. Membrane transport. For things to go in and out of the cell, which is the basic unit of life, that's what's happening. Yeah. Yeah. The key is a hormone, right? The keyhole is the receptor. The hormone is going to go into the keyhole. It's going to go into the receptor. Okay? Questions? So now we got to worry about things going in and out of a cell. Well, certain things could pass right through there. Fatty things, nonpolar things could pass right through there. But there's certain things that are water soluble that can't. Right? To go through, I always use the Lincoln Tunnel. All right, New York, New Jersey, New York. I'm from New Jersey, so I can do that. But going from New Jersey to New York, you have the Lincoln Tunnel. There's other tunnels there too. All right? Boats can go right across the Hudson River to go from New Jersey to New York. They don't need a tunnel. They go right through there. Those are facts. They go right through there from one side to the other side. No problem. The cars can't do that. They have to go through a tunnel, a channel. And that's where the Lincoln Tunnel, the Holland Tunnel, George Washington Bridge, and so forth hold. Well, those tunnels are channels, and they're made of proteins to allow those things to come in. It also helps to turn on the activity of nerves and muscles. We'll talk about those in other courses. All the enzymes, no exceptions, are proteins, and we've got thousands of them. If you don't have enzymes, you'll be dead. To help recognize other things. We won't talk about much about antibodies, but it recognizes them, markers. If all the houses on your street are green and have a picket fence around it, and I tell someone, yeah, just come to my street, my house will be there. There's a green one with a picket fence around it. They're all like that. But if I put a pine tree, a marker in front of my house, and I say, yeah, mine's the only one with the pine tree, that's a marker. That's made out of protein. I can go and look at that. So it's recognition. Recognize that something's there. Movement. The ability that certain things could change shape, like an amoeba and stuff. It could change shape. I'm sorry, movement. Be well, that's one thing. But you could also think of, let's say, muscles. To change shape, right? Short muscle, long muscle. Cell adhesion. Proteins are used as glue to keep the cells together. So you can see there's a lot of reasons why we got to have proteins in there. And we'll talk more about these things if you're really interested. We'll talk about it probably starting next week when we start getting into the cell. Now, the other thing too, this is usually pretty simple for students to understand. Proteins are structurally organized in certain ways. We call it the primary, secondary, tertiary, and quaternary sections. So we have different things here. And this is the way I do it. It's pretty easy. All right. The primary is going to have a long chain of amino acids. And this could be alanine, and this could be cysteine, this could be another cysteine, this could be thymine, this could be methylene, this could be, I don't know, another methylene, this could be another cysteine over here, and this could be alanine over here, whichever, it goes on and on. That order of the amino acids is the primary structure. Okay? Then you take this and you start doing, and let's say I'm just going to draw like a line here, it's not like two E's again. So that's our primary structure. Our tertiary structure, and think of this like a slinky, a straight wire, but now we're going to turn it into a slinky. That's our secondary structure. Okay? Our tertiary structure is taking this slinky and bending it. And believe it or not, portions of that are held together by, what do you think? Hydrogen bond. Okay? So that's a tertiary structure. The quaternary structure may or may not be there, but if it goes, it'll be something like, there's one, and we can have 4 chains put together here. And they're all being, and creating this one structure. That's the quaternary structure. Doesn't mean that there has to be 4 chains there. You think quaternary means 4, but there could be only 3 chains or 6 chains. And if I add, let's say, an iron to the center of each one of these, then I just created a huge protein molecule called hemoglobin, which is in your red blood cells, and each one of those iron centers attaches to an oxygen, and your red blood cells carry oxygen from one part, from your lungs to all your tissues. All right? Questions on that? Oh, yeah. Look at this one. So it's primary, secondary, tertiary, quaternary. Okay? It's kind of showing you up here too. We're trying to, right? Primary, secondary, tertiary, and then they're trying to show you 4 things are in here, and that's quaternary. And again, the way this bends changes the shape and will then define its function. If it bends in a weird place, in a weird way, it's not going to function right. We are going to talk about that. If this is hemoglobin, it's supposed to carry oxygen. Yes, in sickle cell anemia, they're bent in a certain way so oxygen can't bind on there properly. So amino acids can be used more than once to make different proteins. As much as the 26 letters in the alphabet, those 88 keys on the keyboard or the piano, you know how many songs you can make out of that, or revamp old songs and make new songs with just the same 88 keys, or the same 26 letters of the alphabet, making all those millions and billions of words. But the precise sequence of the amino acids determines the protein. You can't mix these up. They have to be precise. Amazing how that does that. But it's all in what we call a genetic code. And I'm not going to go into the genetic code with you guys, that's later on, but I just want to introduce you to very quickly what I mean by they have to be spelled the same way. If those amino acids are not put in the same order or the correct order, it's going to do something totally different. Or maybe nothing at all. For instance, let's say the protein is three amino acids together, with 3 amino acids long. And I'm just going to make it easier for you by just using letters instead. If the amino acid is supposed to spell that, what is that? Okay, all right? 3 amino acids. But what if this amino acid was changed to this by just one letter? Just one letter, the image in your head is going to be totally different. Right? Even if if I add something to, if it's supposed to be that, and I add something to that, it changes altogether. This is almost what we're going to learn later on called an insertion. Or if they're reversed, you take this and it gets converted to this. A totally different word by just changing one thing. It has to be precise. And we have this genetic code to make this. Things do go wrong. They're called mutations. But we have a proofreading system to fix these things. But sometimes they can't get fixed, or they just miss it. And then it gets passed down generation to generation to generation. Where diseases come in. Insulin. Right, you've heard of insulin, diabetics, right? Insulin is a hormone. That's really a protein. It's a protein hormone. And it's made up of 51 amino acids long. Each amino acid has to be in that particular order, whatever it is. If not, you're not going to make insulin. Red blood cells. These are red blood cells. These are what you want. They kind of look like Cert's candies. But with sickle cell anemia, a lot of the red blood cells turn into this sickle, or this crescent kind of moon shape. The hemoglobin, as I showed you before, is made up of 4 chains. And they have to be precise. Each chain is about 150 amino acids long, making it almost 600 amino acids for the whole hemoglobin molecule. Every one of those have to be in the right order. So what is sickle cell about? All of you heard of sickle cell? Yes? Okay. This is what sickle cell disease is, if you understand me so far. A single amino acid is replaced in one of the chains of the hemoglobin. That's it. One. Glutamic acid, or glutamate, is replaced with valine in the 6th position of this chain. That's it. That one difference out of nearly 600 amino acids makes the red blood cells turn into that. And that gets passed down to generation to generation to generation. And I'm going to show you how to predict that in the offspring. Whether you yourself or someone you know, is sickle cell trait or sickle cell disease, or just know about it. We could predict that. So the shape of the protein, as I said, it influences what its function is going to do and what other molecules are going to do around it. If you denature it, it's going to get destroyed. Now, certain things will destroy proteins. They're very easy to destroy. Heat will destroy it. Again, you take an egg. Now, eggs are good. There's a lot of protein in there. Not the yellow. That's a little bit of cholesterol. But the white, or the clear part of the egg, is albumin. It's all protein. When you put that protein, that egg, in a frying pan and you heat it up, you see the clear stuff turning into a solid? It changes the structure. Jello, that's basically protein, collagen. You take it and you put it in the stove, or you heat it up, and you put it in a freezer or the refrigerator, it changes shape. So temperature is going to change the proteins. Acid, our basic body, is going to change the proteins. This makes sense now. Think of if your body temperature, it's normally 98.6, but now you've got a fever, and now you have 101.4. Your body has increased its temperature. You've got thousands of enzymes in your body. That temperature is going to start denaturing, destroying your enzymes. So what's the result? You don't feel like moving. You feel horrible. Because all those enzymes which were supposed to do things throughout your whole body can't. It's supposed to break down foods. You can't break down the foods. So now you don't have that energy. Well, what about all the fat that I have? Well, you got to break down the fat to get it into your bloodstream. Those enzymes are not working well. So we can't keep you at a high temperature for a long time. That's what Tylenol's for. And it makes you feel better. Okay? Make sense? You always got to keep that temperature back to normal.